Alright, so here's our lecture on hematology, or the study of blood. That would be spelled hematology. Anytime you see the root word hema, it's referring to blood. So in the PowerPoints, which you've hopefully read before watching this, because this is just kind of going to give a quick overview of blood, you would have seen that not only is there blood, that's in a blood vessel, various blood vessels, whether it's an artery, a vein, a capillary, an arterial, or venule, and whatever type of blood vessel this blood is in, that blood vessel will be surrounded by certain layers. There will be a layer of what are called endothelial cells. These are a simple squamous epithelium, so they're very, very thin, just acting essentially as a lining for the blood vessel. Then outside of that, you'll have some smooth muscle. Maybe if it's an artery, there'll be an elastic membrane here. Then outside of that, you'll have some connective tissue, maybe another elastic membrane, and so on and so forth. You'll have varying degrees of these layers around the actual lumen of the blood vessel. When I say lumen, I mean the center, like what's inside the tube itself. Now, if it were a capillary, all you would have would be the endothelial cells and nothing more other than a basement membrane, and then there'd just be the tissue right outside of that. No smooth muscle, no connective tissue, no elastic membranes or anything. But the bigger the vessel, the thicker the wall layers, which just makes sense. Now, in the blood itself, you'll have various things. You'll have the plasma, that up here. And the plasma is just the fluid component of the blood. It's the water part of the blood. So that's what everything is suspended in. All the things that are floating in the blood. So cells and proteins, all sorts of things would be floating, suspended in the plasma. So what is in the plasma floating around consists of plasma proteins, for one. So these are just proteins that are oftentimes made by the liver to do various functions, such as albumins, which help with osmolarity, or whether water moves in or out of the solution. Globulins, which have to do with immunity. These wouldn't be made by the liver; they would be made by B cells, which are a type of white blood cell. Fibrinogen, that's another liver protein. You'll see this when we get into clotting, which is called hemostasis. Not homeostasis, but hemostasis. Angiotensinogen, which we just learned about, is a liver protein that gets converted by renin into angiotensin 1, etc., etc. And then all other types of proteins, like all the different hormones that are made of proteins, would be floating around in here. The transport proteins for some of those hormones, like the thyroid hormones, have transport proteins, or steroid hormones. Various other sorts of things like that floating around in the blood. Then we have what's called the formed elements. And these are things that are cells or parts of cells that are going to be present in the blood doing various things. So for one, we have red blood cells, or erythrocytes. And these are the cells that have the job of carrying oxygen, essentially. They have hemoglobin in them, and oxygen binds to hemoglobin. Hemoglobin can be abbreviated as HB. They also have the job of carrying carbon dioxide on the hemoglobin, which we'll see in a couple of weeks how oxygen and hemoglobin and carbon dioxide kind of interact. And inside the red blood cells, we also have the whole bicarbonate buffer system happening, which we learned about in our acid-base balance lecture. Another formed element would be our white blood cells, which are leukocytes and lymphocytes. We'll get way more deep into especially the lymphocytes with our next lecture on immunity. But all of these are white blood cells and responsible for immunity. They help to protect against pathogens. Leukocytes are for non-specific immunity, which means they protect equally well against any pathogen. They don't have to be trained to be responsible for a particular pathogen, which is different from how lymphocytes are. These are for specific immunity, where they have to be trained essentially or programmed to be responsible for one very specific pathogen. So this would be, say, when you say you've become immune to something, you've been exposed to it, you've developed the T cells and the B cells, which are lymphocytes, that are able to protect you against that particular pathogen. So like you get a particular cold virus and you're sick the first time, then you develop the lymphocytes and so T cells and B cells that are specific for that particular cold virus, and you're protected against it. It's also how vaccines work. You train lymphocytes with a vaccine to be effective against a particular pathogen. Also part of the formed elements would be something called megakaryocytes. Megakaryocytes are great big cells. You can see how big they are here. So here would be the red blood cells. A little bit bigger than that would be the white blood cells. And then here we have our megakaryocytes, which are really big cells, and within them are platelets. So these megakaryocytes can release the platelets, and platelets are important for clotting, which you probably already know. Okay, so let's look at our erythrocytes. We'll look at each of the formed elements in turn. So our erythrocytes, they have no nucleus, they have no mitochondria. So because they don't have a nucleus, they can't reproduce themselves. You have stem cells, which we'll get into over here in a minute or two, which are responsible for making new red blood cells. But the erythrocytes themselves are not going to divide and create new ones. Because they don't have mitochondria, they're not super good at making energy either. So they can do glycolysis, but they can't do the Krebs cycle. So they're not going to last a long time because of this. They're going to live about 3 months maybe, and then they go through what's called hemolysis. And that's where they just die, and they rupture, and their component parts are broken down and recycled into new red blood cells. Now inside an erythrocyte, you have hemoglobin molecules, Hb, and this is what carries the oxygen, and also carbon dioxide to an extent. Mostly oxygen, more oxygen than carbon dioxide. Now, to make erythrocytes, we go through a process called erythropoiesis. The word poiesis, it just means to make, and erythro is referring to erythrocyte, so it literally just means to make erythrocytes. This is stimulated by EPO that we would have learned about in the endocrine system slides. So erythropoietin comes from the kidneys, just as a review, and it is produced when you have low oxygen in your blood. So you want to make more red blood cells if you have low oxygen, so that you can hopefully carry more oxygen more efficiently. And these are a type of what are called myeloid stem cells, which we're going to get into in a minute. So for the hemoglobin, this is my artist's rendition of hemoglobin. There are 4 protein chains. 2 of the chains are alpha chains, 2 are beta. You don't have to identify which one is which, but just know that there are those 4 different types. And at the end of each protein chain, you'll have this situation where you have a heme unit and then an iron bound to the heme unit. And then onto the iron, that's where oxygen can bind. So each hemoglobin can bind 4 oxygens there. Okay, so before we get on to the stem cells, let's look at our leukocytes. These are the types of leukocytes. Monocytes and macrophages, these are the same cell type, but it's just where they are. So they're called monocytes when they're in the blood. When they leave the blood and they go out into the tissue, they're called macrophages. So all white blood cells can do this, by the way, leave the blood and go out into the tissue. So let's say the white blood cell goes to the side wall here and it kind of sticks to the endothelial cells. That's called not diapedesis yet, I'm jumping the gun. It's called margination. Like it's going to the margin of the blood vessel. And then it can wiggle its way through the endothelial cell and get out into the tissue. And that little wiggling movement there is called diapedesis. Sorry. Okay, so diapedesis. So they marginate, then they diapedese. right through between the endothelial cells. So they can all do that. So for our leukocytes, we've got monocytes, macrophages, wherever they happen to be, neutrophils, eosinophils, and basophils. All of them can do phagocytosis, so engulfing a pathogen and digesting it. They can all do margination and diapedesis. Looks like I always have trouble spelling that word. Chemotaxis. Chemotaxis just means that they are attracted by certain chemicals. So those chemicals could be cytokines, which that word's written right there. Cytokines are chemicals that are given off by white blood cells to communicate with each other, and sometimes they can attract other white blood cells. There are also chemicals given off by injured cells that attract white blood cells. And then another thing they can all do is amoeboid movement, which means that they can extend a little part of themselves and then kind of drag the rest of their cell body along behind it and just sort of like glomp along like a little amoeba would. Which, if you want to see what that looks like, you can look on YouTube and just see amoebas moving around. They just sort of, they're not that exciting. They just kind of glomp through. Now the production of white blood cells, if we're thinking of leukocytes, so the production of leukocytes specifically is called leukopoiesis. That's stimulated also by EPO because it's going for the same sort of stem cell, the myeloid stem cells that were responsible for erythropoiesis. So EPO just blanket stimulates the myeloid stem cells to produce leukocytes and also erythrocytes. But cytokines, various cytokines coming from other white blood cells can also stimulate leukopoiesis from those myeloid stem cells. Now our lymphocytes, these are the T cells and B cells, which are for specific immunity, and also natural killer cells. These natural killer cells are not specific immunity. They're lymphocytes, but they're not specific, so they kind of straddle between the leukocytes and the lymphocytes. But for the T cells and B cells, these are the acquired specific immunity, and the production of any one of these types, any one of those three types of lymphocytes is called lymphopoiesis. And this is going to use a slightly different type of stem cell, which would be called a lymphoid stem cell, not the myeloid stem cells that we saw for leukopoiesis and erythropoiesis. Now, these lymphoid stem cells are stimulated by interleukins, which are a type of cytokine, and insulin-like growth factors, which we learned about in the endocrine system. Now, for our lymphoid stem cells and our myeloid stem cells, both of these are types of hemocytoblasts. So this is a big word, but this word tells you exactly what it means. We know blast cells. Think back to AMP1, like a fibroblast or an osteoblast. That's something that builds or produces or makes something. Cyto means cell. Hemo is talking about blood, so hemocytoblast is a blood cell maker, essentially. So these are stem cells that make blood cells. So we have the myeloid stem cells. They make red blood cells, and they make the leukocytes. And then we have the lymphoid stem cells, which make just the lymphocytes. Now, both of these are multipotent stem cells. Now, what that means, if you don't remember that from AMP1 or if you didn't hear it in AMP1, if we go down here, multipotent stem cells have to do with the stage of development of stem cells, essentially from the time you're a zygote to when you're born and then so on for your life. When you're a zygote, your cells are what are called totipotent. These are cells that haven't committed to a lineage yet. They can become anything. They can express any other sort of gene. They can make any other new type of stem cell, which just makes sense. You're just a little clump of cells at this point, and you need to make all the different cells of your body, so these things have to be able to do that. Now, as you move further on into fetal development, these cells start to specialize a little bit, and so they kind of grow up, if you want to think of it that way, into pluripotent stem cells. These can differentiate into various types of cells, but they can't make new stem cells, and they can't just completely switch gears, if you want to think of it more in simple terms. Like they're not completely committed to a lineage yet, but they're starting to commit. They're starting to narrow their focus a little bit. And when I say a lineage, I mean a very specific type of cell. So we'll see, for example, once you're born, most of your cells will become multipotent stem cells. They are completely mature, they're completely committed to the lineage, which means that if they are going to be a myeloid stem cell, that is all they're going to do. They can't just be like, no, I want to be a lymphoid stem cell now, and start making lymphocytes instead. They're completely committed to this particular lineage. That's what I mean by being committed to a lineage. They have their job. If you want to think of it like in terms of like your college major, like you're a freshman, you're taking a bunch of classes, you don't know what you want to specialize in yet, you're totipotent. Then you start thinking, okay, I want to get into the healthcare field. Not quite sure if I want to go into medical or radiology or nursing or whatever. You're narrowing your focus, but you're pluripotent. And then once you're fully committed, say you're going to apply to the nursing program this fall, you're multipotent. Okay. So most of our stem cells, like the hemocytoblasts, are going to be multipotent. And only a few years ago, we used to think that they were all multipotent. But just not that long ago, we learned that the stem cells that make the megakaryocytes, which are responsible for making platelets, are actually pluripotent stem cells. And I wouldn't be surprised if somebody finds a few more as time goes on, because things are always changing. So always be ready to learn new things about anatomy as people discover more stuff. But let's look at those megakaryocytes for a second. These are the big cells, remember, that make the platelets. They're pluripotent stems, or they're stem cells that are responsible, I should say, for the megakaryocytes, are pluripotent stem cells. And they make megakaryocytes through a process called megakaryocytopoiesis, which if you just look at poiesis, means to make, megakaryocyte means to make megakaryocytes. So megakaryocytopoiesis are made by these pluripotent stem cells. And then once you have the megakaryocyte, they make the platelets inside themselves. They can release them on something called a platelet ribbon, which is where you have the cell and you stick out this little pseudopod and it's got little platelets on it. And then you just like let that pseudopod go and all the platelets are released out into the blood. Or if the cell just dies, goes through apoptosis, all those platelets inside are just released. So those are the ways that you can release the platelets. Okay, now for our white blood cells, we're just going to get a really quick introduction here before we go into an actual immunity lecture, which will be our next video. But for our white blood cells, whether they're leukocytes or lymphocytes, we can divide them into two categories called granulocytes or agranulocytes. The granulocytes have little lysosomal granules inside them that are visible under a microscope when you stain them. Lysosomal, they're just granules that that cause lysis of a cell, that kill cells. This is where the word Lysol comes from. Antibacterial, antiviral sorts of things. The agranulocytes have other ways of killing the cells, not with those lysosomal granules. They'll just have cytotoxic enzymes, which both of them have, to kill viruses and bacteria, so what we think of as pathogens. So these granulocytes look grainy under a microscope. These agranulocytes do not look grainy. So for the granulocytes, there are neutrophils. These are just essentially generic phagocytes. They are the type of white blood cell that you have the most of. They're the first to a sign of invasion, and they are particularly drawn to phagocytize antigen-antibody complexes. What that means, an antigen-antibody complex, means that you have a cell, here's a virus right here, so V for virus, and it has a name tag on it. And that name tag is called the antigen. And we're going to get more specific about antigens, and it really is very specific. It says exactly what kind of virus this is. It labels it. It's like a perfect little name tag for this virus. But what the immune system does is it will take an antibody, so that will be specific for this particular antigen, and it will kind of stick to it. So the antibody just kind of glomps onto the antigen, and now we have an antigen-antibody complex. And this is just really attractive to a lot of phagocytes. They're like, oh, that looks like something yummy to eat, and they just engulf it and digest it, and it's essentially marking this virus or bacteria, whatever it is, for destruction. Now, eosinophils and basophils, they can both do they can both phagocytize pathogens. They're both attracted to antigen-antibody complexes. But eosinophils can also do something where they send out cytotoxins to kill larger pathogens, like a parasite. So let's say you get a tapeworm in your stomach, and that's just too big to phagocytize. So the eosinophils are going to send out these cytotoxins and try to kill that parasite. Basophils, in addition to being phagocytes, they are the cause of inflammation, or they help stimulate or make more inflammation. And inflammation is not necessarily a bad thing. If you have too much inflammation, it's a bad thing, but you do want to be able to have some inflammation as part of your immune response. And what it just essentially does is it increases blood flow to an area so that you can bring more white blood cells to that area. So the way that these basophils do that is they secrete two things, histamine and heparin, and both of those things cause vasodilation, and they are anticoagulants. So they make the blood vessels bigger, more blood can get through, and they prevent clotting. This also helps as negative feedback for the blood clotting process, so you don't just get a big old clot that gets out of control. Now for the agranulocytes, these include the monocytes / macrophages. These do something called antigen presentation. Antigen presentation is something that is when a white blood cell phagocytizes a pathogen, it can essentially display that little viral antigen on its own self. And that helps to stimulate things like B cells, which we'll get into in the immunity lecture. But it's essentially like taking a picture of somebody and being like, hey, have you seen this guy? And sending these other immune system cells, and usually B cells, out to do something about this particular virus. And they secrete cytokines, which are used to help stimulate lymphopoiesis or to do chemotaxis, to activate B cells into making antibodies, all these different sorts of things. Then we have lymphocytes. We have seen the T cells and the B cells and the natural killer cells already. These two are specific. This one is a non-specific one, and we're going to have a lot more about these two in our next lecture, so we'll just leave that there for now. Okay, so just to get ready for our immunity lecture, I know this lecture is going on a little long too, I'm sorry you guys. When we're looking at that antigen, so let's just look at that antigen in a little more detail. So here's the virus, there's its little antigen. But see how it's got this little, I put this kind of like little crown looking thing on there. That's a specific region. That is the region that says exactly what virus this is. Because all of your cells have antigens, all cells have antigens. So like your cells in your body so this is, I'll just put a Y for you, it's part of you. It has a little antigen, and let's make it look like a little bit different, its specific region. So that when your immune system is going to look at that antigen, it's going to be like, okay, that's us, that's part of me, I'm not going to do anything about this. But when it sees this virus, the immune system can not only know, like, okay, that's a virus, but that is this specific particular type of virus, this particular strain, and it can know what to do about it. And the way that it does it is through the specific region on that antigen, which is called the epitope. So the epitope is the part of the antigen that tells your immune system exactly what this virus is. Now, on a cell like a T cell, say, that is a very specific immunity sort of cell, it can only respond to a very specific antigen. And let's say this T cell has been programmed to respond to this specific antigen. this T cell has something called a CD marker, which is just another membrane protein that sticks out. And it acts almost like a receptor, kind of exactly like a receptor on it. And it's going to be able to read the paratopes of these antigens. And it can do this because it too has a specific region called the paratope. So the paratope reads the epitope. The paratope of the CD marker on the T cell can just sort of fit right in. If it's the epitope that this paratope is specific for, this paratope can fit right in, and that causes a reaction. Once we link these two, it causes a reaction inside the T cell, and it causes it to do various things. Now, if it is not exactly specific, but it's close. So say this comes across a virus that is similar. There's the antigen. Let's make it have just like two little peaks right there. So it's not got these three peaks of this one. So it's a little bit different. It's the same kind of virus, but a slightly different strain. This paratope is not specific for it, but it can sort of kind of bind there. It's kind of close enough-ish for it to bind. And then it can mutate really, really quickly in order to become very specific, to get paratope specific for this new epitope. And that's called cross-reactivity. And this allows your immune system, I shouldn't talk and spell cross-reactivity. This allows your immune system to respond to new viruses, new pathogens, or things that are similar to things you've been exposed to but not exact. Now, last thing to say, or last thing to mention, the CD markers on the T cells. So these things, not only do they have these paratopes, we need to also think about just what they are. They're on a lot of cells. They're not just on T cells, but all T cells have something called a CD3 marker. And then they're also going to have either a CD4 marker or a CD8. So if this is a T cell, there's a CD3, and then it might have, let's say, a CD4. Or it could have a CD8. It won't have both a 4 and an 8. This is going to be important for how they respond when the paratope binds to the epitope. Now, they might not have a 4 or an 8 at all. They might just have the 3. And in that case, they're going to be something called a suppressor T or a Treg cell. And those act to keep the CD4s and the CD8s in check. They keep them from overreacting to things. Okay, so that is the beginning of our immunity lesson. We'll take a pause here. Next video will be all about immunity.